Hey everyone, I'm Jamie and I'm presenting epigenetics and specifically how it works, how its effects are passed on, and what that means for us as humans. So starting with some baseline info, we all know that DNA is the genetic code that builds us and that we inherit the genetic information stored in it between generations. Many factors influence how inheritance occurs, what gets inherited, and to what degree traits are expressed. Some of these include genetic variation from mutation or recombination events, or levels of inheritance possible for traits. Epigenetics is essentially differentiation and a source of genetic variation, affecting the degree of trait expression over time in a cell. And this occurs through a few possible mechanisms. So it's relevant because just like our genome, everyone has an epigenome, and that's how our DNA is told to work. The relevance lies in the effects we have on our epigenome now, and in turn, what those effects could mean later on in a long-lasting way to further generations. Many diseases are caused by errors of the epigenome, such as ATRX syndrome, ICF, Fragile X syndrome, and a handful of others. Beckwith-Wiedemann syndrome, it's a form of organ overgrowth, and various cancers are all possible. Important to note for these that aside from the cancer specifically, all of these are usually found in newborns and young children, and most diseases arising from epigenomic change are intellectual disabilities. Most effects aren't going to be this distinct. They're going to be more menial changes in gene expression, though the important thing to understand here is the causes, especially, because there are significant effects as we listed above. So causes include most of what we do, from diet and its quality to sleep and stress, drug abuse, traumatic events, and even just aging. They all cause epigenomic changes that in turn can later be inherited, and effects vary. In a deeper understanding of epigenetics, it's the changing of chromosomes without altering the DNA sequence itself. And there are three main mechanisms we'll observe: histone acetylation, DNA methylation and demethylation, and microRNA. In each of these mechanisms, the effects, or at least parts of the mechanism, are inheritable. So starting with histone acetylation, so chromatin wraps around histones, which then wrap and fold around each other to condense to form a chromosome over the entire length of DNA. Nucleosomes, more specifically, are the grouping of DNA and histones formed from the DNA that enters through an H1 histone, wraps around an 8 histone core, then threads back out again through the H1 histone. And that process repeats further down. The different histones are variable in conformation. The 8 histones in the core are typically comprised of 2 copies each of H2A, H2B, H3, and H4 histone proteins, but this can vary. Histones can be acetylated by HAT enzymes, histone acetylases, and deacetylated by histone deacetylases, HDAC enzymes. An acetyl group affects a histone's charge, with the group on it turning it from positive to neutral. The amino acid lysine is where acetylation occurs on histones. When an HAT enzyme acetylates lysine's R group, it causes a conformational change of the histone, which in turn loosens the DNA attached to it. This loosened DNA can now be replicated. The reverse is accomplished with HDAC, and returning the DNA into a wound state prevents replication of that DNA because RNA polymerase cannot attach to the tightly wound DNA and histone. From this, we know that acetylation unwinding means an increase of expression of the gene, and the deacetylation is the reverse. So this is a process constantly happening in our bodies, expressing and then blocking genes as they're needed. A study of this here. So this is a study on rats and their transgenerational abilities to process cocaine. There are 2 parent generations over 60 days. One was given cocaine as much as they please to come back to it, and the other was giving saline. After reproducing, both new generations were subjected to either set amounts or self-administration of cocaine, as well as amounts consumed, production of BDNF, or brain-derived neurotrophic factors, mRNA, and protein in the medial prefrontal cortex was tracked. This is how the chemical influence of cocaine is broken down and handled over time. Results showed that the parent generation had no effect on the rate of acquisition of cocaine for any female offspring. Basically, they're all getting dependent on the drug at the same rate. However, male rats whose fathers were sired to cocaine showed a lower maintenance for the drug than the saline offspring. So basically, they were resistant to the cocaine, requiring less of it because their fathers had used it. Figure 3 shows us a female versus male figure. Male's the only one with significance. And figure 2 shows us the same thing, but with the types of administration. And as you might have guessed, the rats that were allowed to have cocaine whenever they wanted tended to have more, a lot more. The resistant males also had higher concentrations of the BDNF factors, and the cause was found to be a heightened gene expression allowed through the acetylation of far more H3 histone proteins, where the BDNF promoters were associated. This can be seen in figure 4 in these two graphs, C and D, showing the association of these factors. From this, the researchers showed that the resistance factor was inherited in the male offspring of cocaine-sired rats, specifically in males. The next mechanism is DNA methylation. So methyl groups are added to adenine and cytosine nucleotide bases of DNA and can act as an off switch for a gene. DNA methyltransferases, or DMNTs, transfer the methyl group and cause inhibition of the promoter area of a gene and block RNA polymerase from binding. Excuse me, potentially blocking the whole thing. This process causes cell differentiation, so blocking unnecessary genes as to make a specific type of cell. In A, on the top figure, we see cytosine being methylated into 5-methylcytosine. DMNT transfers the methyl group from an SAM molecule and leaves it as a SAH molecule. SAM is a very common molecule to be used in this methyl transfer, but it's not the only possibility. There are 3 main types of DMNTs. DMNT1 is for maintaining methylation patterns. So if a cell is synthesizing a new strand and it wants the exact same strand with the exact same methylation, that's what this is for. And there are some parameters for that to occur. DMNT3A and B and 3L are de novo, or new methylation, and it's a lot more lenient in that sense. It can happen anywhere that it pleases, really. In another study, researchers looked at social patterns and odor detection of mice. The parental generation was given odor fear conditioning, so forcing feelings of fear when an odor is detected. And the similarity of offspring response was observed, even though they've never experienced that. And the results showed that many offspring generations responded to a variety of odors with fear similarly to the parent generations, respectively. The inheritance of these traits were also passed differently. If we look at these here, these are the generations along the bottom, and this signifies significance. We can see this was transgenerational, this had no effect, this had a major difference in the parental to first generation, and then completely dropped off and was lost in the second. And then here we see it, the ability to detect and react to odor only appear after the first generation passed, without it. So it's found that the methylation pattern created when cell differentiation was occurring in the offspring gave a stronger sensitivity and allowed for these more unique reactions to the respective odor of the fathers in the parent generations. So what they were subjected to, basically. And it just shows that the methylation patterns can be passed on and influence differentiation further than just telling what kind of cell it's supposed to be. Um, Finally, microRNA. So microRNA doesn't code for proteins. Instead, it bonds to mRNA and blocks ribosomes from translating a gene. This is a method of gene regulation post-transcriptional. You can have everything laid out to create your protein, and it won't happen. The microRNA exists in the cell, kind of like a lipid bilayer, folds itself up, matches base pairs, and just kind of waits for the right proteins to move it to the mRNA, attach it, and then it will do its job of blocking translation. Um, in an organism, the greater need or use of these microRNAs influences how much will actually be produced from the genome, and that can be passed on. If there's a great need for it, it will be seen similarly in another generation. If there's not, then there won't be later on. So to recap all that, epigenetics is the observable change in our chromosomes and genetics, not caused by DNA sequence changes. Histone acetylation is winding and unwinding of DNA to control gene expression, and we observe that in the cocaine tolerance in rats. DNA methylation is for the activation or deactivation of genes for cell differentiation, and we observe that in the odor responses of mice. And finally, microRNA is another form of RNA we produce just to block mRNA from translation, and that's inherited as needed. So all these mechanisms have heritable effects, and notably, a lot of changes can occur from everything we do. And again, whether it's our diets, our stress, what we've been through traumatically, our fear of smells, or the amount of cocaine you do, it's all going to affect our epigenome, and that will be passed on as well. So with that, here is my works cited.